Buckle up, it's a lot of slides, and I want to start off by saying thank you to my team, my present team, to past team, as well as collaborations with the likes of Evosep, Bruker, Thermo. It's been a toil over the 2 decades to get us to where we are at the moment. And hopefully through this presentation, I'll convince you that we are there, and proteomics indeed is empowering drug discovery research and as such. So with that, without further ado, I'll move on to the next slide, and let's talk a little bit about. I don't know how many in the audience love biking. I certainly do. And what hit me about this quote from Keith Bontrager, who founded Trek Bicycles, was he usually used to say, pick 2 out of 3. And this is essentially how we did proteomics for a long period of time. And this is the analytical challenge that we deal with the proteomics pyramid. So we always said, well, pick 2, we can't give you all 4. We can't give you depth, sensitivity, quantitation, and throughput. And that's changed. It's taken a long time to get us to the point where we are at, and I think it's changing rapidly even further. But I can say confidently, and hopefully you will see it through my presentation, that we are able to get our hands around this this analytical challenge that is the proteomics pyramid. So with that, let me move on and talk a little bit about the push and pull associated with innovation versus standardization. And I think Evosep, I would give them credit for really being absolutely adamant about about standardization, and for good reasons. And I think as I've gone through the years, and I've had this push and pull between, do we innovate or do we standardize? And I think I came across these beautiful quotes from Sukant Ratnakar as well as Daiichi Ono. Daiichi gets credited for the Kaizen principle that he introduced at Toyota, but I think those really speak to that back and forth between innovation and standardization. And so again, another quote I found was a good one that's at the bottom of the slide on the right-hand side, which says, we shouldn't be looking at standardization and innovation as opposites. They're not. And if you really think about it as you work in the labs, that you typically get into a habit of doing certain things, and through that comes innovation. So because that is standardization, and then from that standardization, you get innovation as well. Otherwise, without standardization, it's a chaotic performance. We've seen that time and time again, be it nanochromatography, be it mass spectrometry methods, and so on and so forth. And I think this Kaizen principle really does work. I mean, you could, and one can then move this massive challenge or Hercules, as you would call it, or this big ball up that incline, but you have to put some of these standard workflows in place to make sure that you move up that. So I hope I'm convincing you that this is important, and we have to look at both sides of this coin. So with that, let me just touch upon, I think you will see through my talk that there's incredible versatility to deliver biological insights across the drug discovery process. And proteomics is now being applied all the way from target identification selections to lead discovery optimization, preclinical studies, be it for biomarkers, tox assessments, or so on, and well into the clinical studies as well, from exploratory for markers of response efficacy, and so on and so forth. So with that, the question that we ask ourselves every day is, are we enterprise ready to deliver biological insights into disease biology? The cloud of doubt over MS-based proteomics has been around for 2 decades, and this word cloud captures that. We've all heard that multiple times over. How do you clear that cloud of doubt that's over this MS-based proteomics approaches? And it's been a steady march to clear that doubt. And it comes about from innovating, transforming, standardizing, and repeating that, rinse and repeat, and continue doing that. So there are 3 major steps to this. It's automation, so we can simplify to achieve rigor and enhance throughput. improved instrumentation for deep coverage with quantitative reproducibility and scale that will provide reliable performance time and time again, day and day again. And having the appropriate data analytics, embracing AI/ML-based innovations, modularizing the pipeline components, and incorporating cloud computing to achieve that scale. So let me touch a little bit upon next-gen capabilities for differential protein expression assessments. And what you see here is a bubble plot. On the x-axis is the year, and on the y-axis is the proteome depth. And this, okay, that's proteome depth. And so what I want to show you here is going between, say, 2017 to present day, the proteomes per MS day has increased by about 17x. Sample requirement at the same time has dropped by 13x over that same time frame, and the percent data without missingness has improved from about 45 or 55% missing to about 10% missing now. So 90% of the data is. So that's incredible achievement to make that happen over a short period of time. And this is almost now a year old. What's happening internally within the team is further along than this as well at this point in time, though I'm not at liberty to speak to it today. So with that, create voluminous amounts of data. So you need scalable cloud compute infrastructure to be able to do that. We've embraced deep machine learning approaches so we can organize, integrate, and leverage the AI/ML to analyze these large data sets. And the question that you have to ask ourselves, well, why do all of this? And the answer to that really is so that we can enhance our phenotypic and nodal screening from HTS full deck screen to deconvolute new targets, accelerate medicinal chemistry, be it selectivity profiles, off-targets, pathway assessments, proximal markers, enrich our target biology so we can understand MOA better. Target engagement studies can be done, preclinical studies. Of course, I'll touch upon translational efforts, and you'll see some of that. And another place that I think is close to my heart is to elucidate immunogenic hotspots to assist with therapeutic antibody engineering as well. And proteomics plays an important role there too. So with that, I think I'll change gears over here and talk about how are we applying proteomics across this drug discovery value chain, starting with screening efforts. And these will be vignettes. I'll stay pretty high level with this, but and then move on to translational activities, and finally touch upon plasma proteomics as well. So with that, this is the typical screening tier that happens across multiple biopharma as well as biotech in a screening paradigm where you start with a full deck screen, a primary screen. It could be a phenotypic screen. You retest, you run counter screens, you perform potency and selectivity measures. And I believe this is where proteomics can start playing a role. We are seeing that time and time again, where we can use global profiling approaches, phosphoproteomics approaches. It could be single point or dose response, and I'll show you some of that data later on. It could be also time course. So that will give us better hits, and those hit evaluations can happen. And eventually, if it is a phenotypic screen, then you'd want to go about doing target deconvolution using chemical proteomics approaches so that you can identify the targets and have biologically validated risks. And in the chemical proteomics toolbox, it could be everything from compressed sets, kinome approaches, PAL-MS, or even LIP-MS with dose response profiling. So let me start off and give you an idea as the scale of this. And this is almost a year or two old now. And this is in iPSC cell derived cells. It was a small cohort at that time with about 68 compounds. But I think those numbers and some of the things that's recently happening within the group are upwards of 600, 700 per month, and touching almost 1,000 compounds that get screened using this workflow that's illustrated over here, which uses preomics followed by Evosep coupled to a timsTOF HT. What does a typical data set look like in this particular case with iPSCs? We're starting here with less than 5,000 cells. The protein groups, it's pretty decent coverage that we get. And what you see is the data set over here, so that we can raster through and select our compounds based on these proteome profiles. One of the proteins of interest and what the individual expression or change in expression looks like across these compounds is shown below at the bottom of the slide. So that was for screening. Here is an example of using compressed set surfer for target deconvolution. Of course, I can't speak to things that we have deconvoluted, but as we were getting this platform up and going and the workflow up and going, this is actually taking the melt curves and compressing that in the heat challenge scale, and then running a dose response profile. And this was done with staurosporin just to demonstrate proof of concept. The plot that you see on the right hand side really captures a compressed set dose response versus EC50. And what's below that's highlighted is stabilized proteins that you would see shift, as you can see for CAMK2D being stabilized with staurosporin. So this platform is exquisite in the depth of coverage as well as what it is able to deliver, and it complements global proteomics approaches, transcriptomics to nominate novel targets. It also doesn't require any derivatization of the chemotypes, as is typically the case with PAL. So it complements PAL-based MS as well. So I'm going to switch gears over here. I'm trying to be mindful of time, so not going into too much detail, but I think protein degradation is a therapeutic modality that's been talked about a ton in the biopharma and the biotech circle. I think that these are wonderful articles by Craig Cruz, Phil Chamberlain, and others worth looking into. And it gives us a modality so we can expand the druggable proteome. So if you do have time, and this is recorded, so you should have access to this, please go read through these articles. They are insightful. And at BMS, as most of you know, this is an important modality for us, has been since about 2015, 2016. So I'm going to take you through how we have utilized proteomics to help with these targets and to build SAR. So with that, here's one example, and this is an early example from 2017. And this is how we were running the SAR typically, and you've seen a lot of these volcano plots. It was typically one concentration that it was run at. This was a comparative analysis proteomics. You run a volcano plot, you then say, okay, well, we are seeing a lot of degradation, and give this to the chemist who would then improve the selectivity, and you run the next series of compounds and say, okay, well, this looks pretty good. However, this created challenges for us because there are these rigid artificial cutoffs that you typically draw, a twofold change plus or minus, and a certain adjusted p-value. And then when things move across those boundaries, you start asking yourself the question, well, what does this mean? The proteomics is highly variable, and we're not able to get reasonably good data sets and so on and so forth. So with that, given the power of the platform and the fact that we can run at scale, we started saying, well, why not we just run a dose response profile? So at that point, we started running whole proteome dose response profiles, and what you see here on the right hand side is a dose response profile matrix for one of those compounds. And this is a sliver of the data. There's about, sorry about that. There's about eight compounds here, and a sliver of the data, about 10 proteins. And what you can start seeing, and each one of the proteins has a dose response, an 8-point dose response. And you can start seeing that clearly the target is labeled in green over here, or highlighted. And then you've got off targets that you can see that show up only for certain compounds, and it's flat otherwise for the remainder of them. And then there are some pathway effects that you start seeing as well that are beyond the target. So it's a huge matrix, as you can see. I mean, you probably read the bullets below, so I won't beat through them. But I think this starts getting at, providing a wealth of information to our chemists so that we can start building a robust SARs early on in the program. So with that, just for the sake of presenting, what we started doing was, so we can talk about it more freely rather than thinking about our targets and trying to get clearance. Here is a degrader proteome dynamics, and this is for GBET1. And what you see here is a 24-hour, dose response profile matrix. So now these are not volcano plots. I'm going to point out that this is actually percent degradation on the x-axis and an effect size. So each dot there is a dose response profile. Quite elegant how my past team and some of the members from my current team pulled this together. And each one of those, so GBET1, as you know, will degrade BRDs. So you can see excellent degradation of BRD2, 4, and 3, and you see the dose response profiles below. The proteome is pretty deep at about 9,300 proteins that were evaluated quantitatively. So that's the 24-hour profile. So we said, well, let's run a time course as well. And here's what we see with the time course. So the 2-hour profile is here, and all you see is a clean profile with just degradation of BRD2, 3, and 4. At 6 hours, there's some things starting to come in, and so those are most likely either late degraders or there's pathway effects that are creeping in. And by 24 hours, you've got a combination of and an exploded volcano, if I may call it that, and mostly pathway effects there. So you can start thinking about it from a systems pharmacology perspective at this point. So here's what pulling out one of the pathways, and this is the RNA polymerase II pathway that was pulled out. And what you see here is the profiles for each of these compounds that's in the bubble plot below. But what you see is BRD is an early responder, so that's a degradation event. There is something here with TXNIP, which is a delayed response. You start seeing a dose response, actually a stabilization that's happening at 6 hours. And then there are some late responders, and most likely those are the pathway effects. So there's no change. There's some change at 6 hours, but then you start seeing a beautiful dose response at 24 hours for the HECT D1. And so there are many examples like this. This is deep with 8,000 proteins. So then, I pulled out some things that are in the literature with regard to BRD degradation, the ion uptake and transport protein pathways, and you can see some beautiful profiles there, and you can see how the networks are affected and the pathways regulated, both with the DBET1 as well as with MC1 as well. So I hope that gives you a flavor for how much information one can get and how much biology one can uncover or pharmacology one can uncover with these kinds of approaches. So with that, I'll move on to enabling translational research for generating marker hypotheses. And the team here does everything from early discovery program support. So that's what we call as forward translation. So we do biomarker discovery in vivo and ex vivo models, genetically defined models, and so on. And then translate those markers into the clinical cohort. There's a reverse translation that work that we do as well, where we would, through collaborations or otherwise, get access to clinical cohorts, do marker discovery over there, and bring that back into our either human ex vivo samples or other mammalian models and test out those markers in that. And I'll share some of the examples of that with you. So let me start with the first one here. I think this is an elegant example of using global phosphoproteomics to identify PD markers. So PTPN2 is a central regulator of interferon signaling, which thereby facilitates tumor survival in inflamed tumor microenvironment. And this has been pursued by a lot of companies. There are dual inhibitors of PTPN1 and 2. that are in clinical trials. There's a couple of assets from Calico AbbVie that have been in clinic since 2020. And there were internal efforts here at BMS just as well. When you go into the literature, you look at the PTPN inhibition markers, and typically phosphostatin-1 is what is exhibited. However, the window on that particular phosphorylation event is pretty narrow, so we took on this global profiling effort. And what you see here is phosphorylation changes in a dose-dependent response manner for PTPN inhibition. This is a phospholipid signature that you see, and this is tyrosine phosphorylation events and exquisite dose-response profiles for those. And the data is about 20 to 30,000 phosphopeptides deep. And it was all mined. Here are some other tyrosine kinase phosphopeptides that are showing exquisite dose response. So this starts giving us rich information on PD really early on that we can utilize effectively as the programs move forward. So we didn't stop there. We did kinase inference analysis as well. On the left-hand side, what you see is looking at PTPN 1 and 2 knockouts in cellular models and the kinases that are being impacted. And on the right-hand side, the tyrosine kinase substrates that increase because you're inhibiting a phosphatase, you'll see an increase. But there are some decreases that you see as well for phosphocyanine, and those are pathway effects upon inhibition of PTPN1 and PTPN2. So a lot of information here. It's deep. It's taken, I've summarized a toil of almost 4 months into a slide. So there's a lot of depth of information here. And you routinely applying this. So let me move on to some clinical examples. This is published in Journal of American Heart Association, so feel free to go through it. What I want to really call out here is it's a urine proteomic assessment to look at HFpEF cohort. And this trial has been around since 2014. There was plasma proteomics done using a SomaLogic platform, and then the urine proteomics was done with LC-MS-based approaches. We're talking about a study population here for 426, and only those enrolled at baseline, were analyzed. It was all done on an Evosep 1. At the time, it was on a Q-Exactive HF. This is old data. But we were able to get pretty deep urine proteomes at about 2,300 proteins deep. And what you see there is some really nice data sets that came about. And by the way, this is not a typical volcano plot. It's a hazard ratio versus p-value. And I'm not going to go into detail, but the paper is out there, so you can go take a little read. But we found some excellent markers here, and some of these markers have been utilized now for clinical trials that are ongoing for mavacamten, I believe, which is already in the clinic. So hopefully we'll see some data sets come out of that from these marker sets that were identified here. So with that, I'm moving on to plasma proteomics and give you a flavor for. So we've been toiling at plasma proteomics since the early 2000s, and this is a study. It's a oncology study from 2006 where we utilized plasma proteomics, and at the time we used to call it ion mapping, which is now called DIA. So there's a patent put around this, and we utilized that workflow to run plasma proteomics on a clinical cohort of about 108 patients. And we were able to identify some very intriguing marker sets. And what you see at the bottom of that heat map is actually individual peptides that are showing a difference between the different complete responders versus partial responses versus stable disease at 27 weeks with that particular clinical trial. So we've tried to do this over the years. What is in the middle is a personal communication with Matthias Mann. And that was the next jump from where we were at in 2006. And then we followed that workflow further. We optimized it over the years. And what you see on the right-hand side is a 2018 clinical cohort, and that cohort now it's about 800 patients. So we went from 100 in 2006 to about 800. That's also intriguing data. I can't go into more detail, but we were averaging about 400 to 500 proteins deep with those workflows at that time. So I think with Evosep, with all the standardization of workflows, with utilizing these Magres beads, this strong anion exchange beads, I think at last we are achieving the depth that is required to be meaningful and have the specificity to boot as well. So here we did a comparison. This was done last year of other beads that will go unsaid over here. You guys can figure it out. The SIX beads on our Kingfisher platform or the OT2 Evosep approach, top 4 depleted plasma or neat plasma as well. And various throughputs were utilized so that we knew what the depth of the proteome was. I think it's pretty amazing that we are as deep as we are with about 3,000 proteins deep over here. And also the sample input is very low. We've gone from using mils of material to with the SX beads down to about 4 to 5 microliters of plasma that we start with. We've done that with serum as well, and in this case it's mouse serum. But we've been able to achieve a scalable end-to-end automation that accomplishes deep plasma profiling. And this is just to show you 6 donors over here. These are, I don't know how healthy they are. I don't know if I'm one of them or we've donated over here and generated some of this data. I think it's pretty deep at 3,000 plus proteins. This is using 4 microliters of plasma and consistently getting some very tight CVs as well. So we are very pleased with this, and this is almost a year old now, and things have improved from where these are at this point. So I'm going to leave you here with a couple of last concluding slides. So clinical plasma proteomics has come a long ways. We've finally turned that page, and I think this bubble plot kind of captures that effectively. In 2006, I mean, again, this is a plot of protein groups versus the year. MS days required to execute a study, and I showed you that study. It was 60 plus days to do that, and it's incredible how quickly we can now raster through and complete large studies, right? So the study that we did recently, and those numbers right here, took us about 12 days to run that. And the cohort size was about 1,200 patient samples. So this is the latest, and this year would probably exceed that to probably 3,000 patient cohort, and hopefully with even faster speeds and throughputs without sacrificing the depth of the proteome, we might be able to do it in 4 to 5 days. So excellent performance that we're getting, but that creates a different challenge for us, interpreting all this information. I love this quote from Sidney Brenner. So the conversion of data into knowledge constitutes a great challenge, and it's front and center for us for future biological research. So I think gone are the days of just looking under the lamppost, which is what we've done with a reductionist approach. And we need to start embracing the power of the white space, because there's a lot of information here and we're barely scratching the surface. So exciting times ahead, and I think the data is robust, reliable, and I think the challenge for us going forward is to mine all of this. So with that, I'll end with the slide and say, the progress continues. I think we're going to get to a point where all this data will be available, be it within a pharmaceutical framework, externally as well from public data sets. And we need to combine all of this data through a knowledge graph framework so that we can have better interpretation. So I see this as what the future state is going to look like over the next few years. So with that, I'm going to acknowledge my present and past team. It's been a lot of effort, a lot of folks. If you see your picture over here, if you're on the call, thank you for doing that. Press your pictures on there, find yourself. And all the collaborations that we've had with Bruker Ion Optics, Proteomics, Evosep, you name it. It's a proteomics village. We are a small community, and the reason we are all doing this is because patients are waiting. With that, I'll stop and hand it back to Nikolai.